Okay, welcome everybody to today's ML for Proteins seminar series. Today, we're super excited to welcome back Jeff Ruffolo. He is a machine learning scientist at Profluent Bio, and today he's going to be talking about some of their new work, adapting protein language models for structure condition design. So we're very excited to have you today, Jeff, whenever you're ready to take it away.
Great. Thanks so much for the introduction. Thanks to all the organizers for the opportunity to share some of our recent work. So the talk is titled Adapting Protein Language Models for Structure Condition Design. Just to start, I'll go through a brief recap of protein language models. So with these models, what we want to do is learn a probability distribution over sequences of amino acids. So for a given sequence S here, with a set of residues S1 through N, we want a model that can help us calculate P of S. The way we do this is we use a class of models called language models. And here the goal is to predict the likelihood of each amino acid given all preceding amino acids in the sequence. We can split this P of S into a set of conditionals shown on the bottom of this with this equation, where we're predicting the probability of each amino acid type at position I given all amino acids leading up to I. And then after we've trained this model, we can use it to compute that exact likelihood of a sequence that we want. We typically parameterize large language models as transformers, where there's two kind of flavors of language models: autoregressive models, like I mentioned on the prior slide, as well as masked models. For either model, we use the transformer, which really has two main pieces. These are the alternating self-attention layers, which for autoregressive models or decoder models allow information from prior residues to proceed up to the current residue I. And then for masked language models or encoder models, these allow information from all residues to sort of flow to all other positions. And then the other piece is this feed-forward neural network that acts as a sort of position-wise processing that the model can learn. For this work, we're specifically going to use the ProGen2 language models. These are a family of autoregressive language models trained on a mixture of genomic and metagenomic data sources from UniRef and the BFD database. These are trained with the autoregressive language modeling task, where again, we want to predict the likelihood of each amino acid given all preceding amino acids in the sequence. If we've trained our language model well, then when we generate from it, just by feeding one amino acid, predicting the next, and so on until we have a full sequence, the set of sequences we generate should look like the natural distribution of proteins that we trained on. Of course, for design tasks, generating a bunch of sequences that sort of recapitulate the distribution of your data set isn't particularly useful. So to steer these models for practical purposes, we really have a couple examples. I'll talk briefly about what most people do, which is fine-tuning, or more of a design by examples strategy, where here the goal is to take a pre-trained language model that knows something about the distribution of all natural proteins, and then narrow its generative scope by further training on a particular set of proteins from maybe one or more families. This is the approach we took for our Open CRISPR project, where here, again, we start with a universal protein language model that's seen many, many proteins, about 500 million in the case of ProGen2. And then we can successively narrow its scope by training on just particular sets of proteins. For this project, we put together a really large database called the CRISPR-Cas Atlas that has about 5 million CRISPR-associated proteins. If we fine-tune on that, we can generate just those families. And then we can further narrow the scope to generate just Cas9s. And this approach works well. You can use it to generate tons of synthetic proteins, even for protein families that have very few natural examples. And of course, some of those proteins will function, in this case, in human cells for gene editing, even exceeding highly functional natural proteins. But the challenge for this approach is that if you want to design for a particular function, you need to first collect a data set of proteins that exemplify that function, which can be hard if you don't have a lot of data in the first place for a protein family that you want to design for. And also it introduces kind of an extra step in the design process, where we really need to collect natural examples to enable the language model to generate. So instead, what we'd like to do is move towards a regime where we condition our language models, or do more of a design by constraint, where we have one pre-trained model and then we tell it certain design specifications that sort of indicate the distribution of sequences we want to get out. And this is the strategy that we took for our proseLM model, which I'll talk about today. First, I'll go through the model and how we sort of endowed a language model with structure conditioning. Then I'll talk about expanding the conditioning in that model to more than just the protein backbone. And finally, I'll talk about some special considerations we had for modeling antibody interactions. Starting with the model, our approach is to use a set of layers that we add into the language model called adapter layers. And specifically, we want to use these layers to inject information from the protein structure itself. Starting from the bottom of this middle panel, we built this network that we called the causal encoder, which serves the purpose of taking in a protein structure as well as surrounding molecular context encoded as atoms, and then producing a set of embeddings that we can feed to the language model. The encoder here takes the form of an encoder-decoder GNN, similar to things like protein MPNN and prior methods for structure-to-sequence generation. But we also use these IPMP layers as well. So you can think of MPNN layers as operating on residues and their neighbors as nodes and edges, and then the IPMP also adds in some more geometrically inspired features for each residue. And then the second piece are these adapter layers that connect the structural information from the encoder with the language model that's going to be generating the sequences. The key principle behind these adapter layers is we want to add in this conditioning information without adding tons of parameters to the language model. This helps us be more data efficient because we're not feeding a bunch of parameters for this conditioning task. And it also helps keep the language model quick for inference, so we can generate many sequences without slowing down the model itself. The way this works is we take, for each layer in the language model, we insert one of these adapter layers, and those layers take as input the language model features at that layer. So this is going to be some high-dimensional vector per position in the sequence. And we also take the structural features for that same residue that we're generating for, which will be some smaller low-dimensional vector for each position. We then take the language model features and down project them to a lower rank representation. So this might be a vector of size 16 for each position. We concatenate that lower rank language model representation with our structure features. We do some processing with an MLP layer just to kind of incorporate that structural information with language model sequence only features. And then we project back up to the initial language model dimension and add that back onto the features with a dropout for regularization. So in total, adding these layers throughout the depth of a language model might add anywhere between 0.1% to the overall parameter count up to a few percent for smaller language models, where the adapters tend to contribute more proportionately. We first validated this model on this Cath 4.2 dataset that's become pretty standard for evaluating structure-conditioned sequence design models. In this plot, I'm showing the perplexity on the test set, just with language models going from left to right now, increasing in scale from ProGen2 small, which has about 150 million parameters, up to the largest ProGen2 model that has about 6 billion parameters. You can see even these models that don't have any structural awareness, that are just looking at sequence, assign lower perplexity as you increase the model scale. So they've better captured this space of sequences, and they can assign lower perplexity when you present them with the sequence in the test set. As we incorporate structure, you can immediately see that having information about the 3D structure of the protein already narrows the space of sequences that the model is considering. So the causal encoder, which is trained just on protein structures, has no language model component, already assigns a lower perplexity to this test set than even the largest ProGen2 model. And then as we combine these components with proseLM, where we have the causal encoder, and then corresponding ProGen2 models with proseLM-S corresponding to ProGen2 small, we see further reductions in perplexity, with the largest proseLM model achieving the lowest perplexity overall. We also want to use these to design new sequences, not just score the likelihood of a sequence given a structure. So to do this, we need to decode simultaneously from the encoder and the language model. So this is just a diagram showing kind of how that works. So on the top, we have the language model, which starts with a start token. In this case, for Persian 2, it's 1. And then it generates the following amino acids one after another. And similarly, the causal encoder takes in the sequence we've generated so far and then produces embeddings that are helpful for generating the next residue. So when we want to design a sequence from scratch, we do these in tandem, where the language model might spit out this Y, and then we'll feed that back into the language model, as well as the causal encoder to inform the generation of the next position. Using the same Cath 4.2 test dataset, we measured native sequence recovery for the causal encoder in gray here, as well as the 4 proseLM models. And similar to before, we can see that as we scale up the language model component of the proseLM models, we achieve higher sequence recovery on this test set of diverse protein structures. And those increases in recovery are really distributed across both core residues, shown on the left of this plot, and surface residues towards the right. So residues at kind of any level of burial benefit from the evolutionary information that comes from the protein language model. We also broke this down by length of the sequences that we're designing for. And here you can see that for shorter proteins, we tend to see lower rates of sequence recovery. These tend to be proteins that have less constraints on the sequence. You might have a higher proportion of surface exposed residues versus larger proteins on the right, where we see the highest rates of recovery. And there's another smaller trend here where you'll see a bigger benefit from larger proseLM models as sequence length grows longer as well. We also looked at the impact of adding noise to the protein structures during training. This is something that was done for protein MPNN, where they showed that if you train on structures with a small amount of Gaussian noise added to the coordinates, you can design sequences that are more robust and more likely to yield confident structure predictions when you run AlphaFold 2 in single sequence inference mode. We find a similar trend for proseLM models, with the causal encoder pretty closely reproducing the results for protein MPNN, where this light gray line is the model trained with no noise, and the dark blue line is a model trained with a small amount, 0.01 angstroms of Gaussian noise added to the coordinates. And on the y-axis, we have the fraction of sequences for our test set that were successfully predicted to fold into the starting structure by AlphaFold 2 using a cutoff of LDDT above 90. And you can see going from left to right here, as we introduce language model components with proseLM and then scale those language model components, both of the lines go down, meaning that the larger language models tend to produce sequences that are perhaps more robust to this noise, and also approach the native sequence rate of successful AlphaFold 2 structure prediction. And just to really drive this point home, if we change this cutoff of LDDT to any other set of reasonable values, we see this trend kind of across all of the different cutoffs we could have tried. So as we scale the language model, we see a smaller enhancement in this successful AlphaFold 2 prediction when we go from unnoised to noised. Now I'll move on to incorporating more context beyond just the protein backbone, starting with some in silico results with sequence recovery, and then moving into some design validation we did for CRISPR-based genome editors. The first additional context we can expand to beyond the backbone is just other proteins. With the encoder, this is really straightforward. We can just extend the graph that's encoding the protein backbone to then include other proteins that are forming complexes with our protein of interest. If we do this, in the plot on the right, you can see going from the lighter blue line to the darker blue line, across all the proseLM model scales, we see a reduction in perplexity. So adding in other protein chains further narrows the sequence space the model considers, resulting in this lower perplexity. If we go further and include things like small molecule ligands or nucleic acids or ions, we again see a perplexity reduction. This time it's smaller because these tend to have a smaller footprint in the structures we evaluate on. But again, you see adding more context restricts the design space considered by the model, yielding a lower perplexity. Here I'm going to show results for sequence recovery when we have either just the backbone provided, those are the bars you see now in the lighter colors, versus all contexts that a given protein backbone could be surrounded by. So this is other proteins, this is nucleic acids, ligands, ions, etc. And for all of the models, we see about a 3 or 4% increase in sequence recovery across the entire design protein chain when we provide this extra context. So the improvements from adding additional context to the model appear to kind of be complementary to scaling the model. Regardless of which model we look at, we see that 3 to 4% increase in sequence recovery. As you might expect, the largest increases in sequence recovery are directly in the vicinity of this added context. For example, in the plot on the left, we're looking at residues within 5 angstroms of nucleic acids. And you can see for all of the proseLM models, you see a sizable jump in sequence recovery from the light-colored box plots, which are just the backbone, to the dark-colored box plots, which are sequences designed with that context provided. And we see a similar trend for ligands as well as ions. We also looked at how the change in sequence recovery sort of deteriorates as the residue that you're considering gets further from that context you're providing. So in these plots, using the first one for proteins as an example, on the x-axis we have various distance bins that a residue can fall into. It can be within 5 angstroms, like the plots I showed on the prior slide, or it can be progressively further away from that context. Then the y-axis is showing a change in native sequence recovery with that context provided versus without. And you can see for proteins, for instance, we see this large increase in sequence recovery up to nearly 30% higher sequence recovery for the encoder model that drops off when you get, say, 10 or 11 angstroms away from that extra context. And you can see a similar pattern for nucleic acids, ligands, and ions. But the differences are kind of interesting. For instance, for nucleic acids, again, we see the causal encoder having the biggest increase in native sequence recovery when we provide the nucleic acids. But as you go further from the actual nucleic acids, you see this crossover point where larger proseLM models tend to have a increase in sequence recovery across the entire protein if they were given nucleic acids versus the causal encoder, which is mostly constrained to that 5 angstrom distance in terms of shaping the entire sequence you're designing. And then for ligands and ions, it seems like most of the benefit in sequence recovery is only in the vicinity of those atoms that we're providing. There's no difference between the different models as you get further away. I was a bit surprised by the large increase in sequence recovery for proteins when you provide the context, because 30% seems like quite a lot. So I looked at the changes in residue burial with respect to sequence recovery for these test case examples. And it turns out the increase in sequence recovery is largely explained by just new residues, residues changing in kind of their burial degree. So here what I'm showing is a similar plot to a while back, where on the x-axis we have a measure of burial, where further to the left means a residue is more in the core of a protein, further to the right means it's more on the surface. And you can see the trend for sequence recovery when we provide full context or backbone only is largely the same. So we're getting that 30% by a residue that was on the surface becoming more buried. But there isn't some large difference here where we're seeing recovery shoot up for less buried positions. Protein language models are also commonly used for fitness prediction. So we wanted to see what the impact of adding structure with proseLM has on the ability to do this zero-shot fitness prediction task. So we took the deep mutational scanning data sets from the protein gym benchmark set and compared Progen 2 models, so just the language model, with 2 sets of proseLM models, either trained on just protein backbones on the Cath 4.2 dataset, or trained on backbones with additional context. This is the PDB darker blue line. And in these plots, the metric that I'm showing is NDCG at 10%. So this is measuring the ability of the language model to rank the highest fitness variance, and in particular, to return the best 10% towards the top of the list, where higher values here are better. And you can see that when we compare proseLM models to ProGen2 models, we in general see a slight enhancement in this ability to prioritize these high fitness variants, though they do start to converge at the largest scale. And then I've provided values for ESM IF1 and protein MP9 on the right to contextualize these results for models trained just on structures. Even though the overall result is that structure tends to help, the patterns that you see for this metric look pretty different based on what type of fitness we're interested in. For stability, for instance, we kind of see an opposing trend where the ProGen2 model gets better at pulling out high stability variants with larger scale, while the proseLM model actually slightly degrades in its ability to pick out these high stability variants as you increase the model scale. And in fact, just the encoder tends to be the best at finding these high stability variants. You also see something interesting for binding fitness landscapes, where here we see this gap open up between the dark blue line, which is the model that was trained to see things like nucleic acids or ligands, versus the light blue line, which was just trained on backbones. Here, it seems like what's happening is if you train the model with explicit context of things your protein should be binding with or interacting with, if you then go to a fitness prediction task where you're expecting it to predict things like binding, it's no longer as capable of doing that if it doesn't see the actual thing it's trying to predict binding for. So it turns out that training without ligands might be useful in some cases as well. Moving now towards some experimental validation, I want to start with Cas9 proteins and just briefly introduce these. So Cas9 proteins are these complex molecular machines. They're used in genome editing applications, and they tend to be challenging to design because we have this large multi-domain protein. All of these domains need to coordinate to carry out their activities. So kind of going around the protein, first we have these nuclease domains. These are responsible for cutting single-strand DNA. We additionally have a set of recognition domains, which need to recognize a particular guide RNA sequence, as well as detect mismatches between your guide RNA and the target that you're aiming for with your genome editor. And then we also have this PAM interacting domain, which acts as a sort of DNA motif recognition domain and sits upstream of these other functional domains as it needs to melt the double-strand DNA so the rest of the stuff can happen. And on top of all of these domains, we also need coordination between them. So like I mentioned, we don't want the nuclease activity to happen if we haven't recognized the correct site, if we haven't melted the DNA based on this PAM interacting domain. And of course, there are smaller details, like certain domains need to coordinate metal ions for cutting the DNA, and so on. So we wanted to see if we used proseLM, this model that can design protein sequences based on the structural context of Cas9 in this case, as well as the context of these nucleic acids that it's binding and cutting. Can we sort of sample the sequence space around a really good genome editor like SPCas9 and find variants that maybe aren't better, but have some different trade-off in their editing activity that could be useful for particular therapeutic or agricultural applications. The way we approached this was we took two structural states from the PDB. One is this binary state where we just have the protein bound to its guide RNA, and the other is this catalytic state, which is closer to its functional form, where it's going to cut the target DNA. We used both of those for conditioning with proseLM. And then we also collected a DMS dataset from the literature, as well as a multiple sequence alignment from our CRISPR-Cas atlas dataset, and fed those in as sort of positional residue biases for design. The reason we added in this DMS and multiple sequence alignment data was kind of empirically driven. If you look at the plots on the right, And the top one, what I'm showing is the Hamming distance of a given design based on what strategy we used. So the light gray is just proseLM with the two structures as input. The dark gray is with the additional information from deep mutational scanning data and multiple sequence alignment. And you can see the dark gray is significantly shifted to the left. So by giving this additional information, the model is producing sequences closer to the starting natural protein. But the benefit of providing the extra information isn't just to kind of stay closer to your starting point. If you look at the plots on the bottom here, I'm showing the perplexity of a given design sequence, given either of the two structural states we used. And you can see that the dark gray here is also significantly shifted to the left, meaning that providing these constraints helps the model produce sequences that it also thinks are better for the backbones. So we ultimately selected from this dark gray portion a set of 9 SPCas9 variants. So these are sequences that are fairly close, but also highly favored by the model. We tested these for indel formation across 3 target sites in the human genome. In general, we found that our variants had a range of editing efficiencies across these 3 target sites. There was one that was particularly interesting, this variant 3, where we saw a significant increase in editing at this HEC2 target site, and then perhaps comparable or slightly degraded editing at the other 2 target sites. So we don't necessarily think this is an all-around better variant of SPCas9, but this kind of showcases the ability of a model like proseLM to sample the native the nearby sequence space around something like SPCas9, which already works quite well. But you might want to shift the activity of for particular sites. Moving away from Cas9 design, but still in the genome editing space, we also looked at these deaminase proteins. So these are proteins that we had previously designed using a similar sort of design by example fine-tuning strategy that I mentioned at the top, where we collected a bunch of natural examples and generated these type A-like proteins. And the reason we want to do this is because we can fuse these to a Cas9 protein to work as a base editor. So instead of double strand breaks, we're now just doing precision single base pair edits of a genomic target. So we tested a bunch of these before. We found a few that were highly active, but of course there was kind of a long tail of low activity base editors that came out of our language model. So we wanted to see if using this structural approach could take one of these weakly active base editors and make it into something that's a bit more functional. So to do this, we built a structural model of the deaminase in its active conformation, just using AlphaFold and homology modeling. And then we targeted design to either residues right next to the DNA that's going to be base edited. These are the red spheres in this structure. Or positions that are further away from the base to be edited. These are the red spheres in the right structure, of which there's, of course, many more positions that can be designed in that scenario. And then depending on which of these design strategies we were doing, we fixed all positions that didn't fit into the allowable positions to be designed. As a brief aside, the way that we fix positions with proseLM, which typically we are decoding sequences from N to C, requires a bit of a trick. So the decoding strategy I mentioned before is this kind of tandem decoding where we decode one residue and feed it back to both models. If we want to design with fixed positions, we can actually just tell the encoder model residues that are yet to be designed. So in this figure, residues I and I+1 are things the language model hasn't decoded yet, but we can tell the causal encoder that these are coming in the future. And then that lets the language model kind of indirectly see the fixed positions. Going back to the base editor results, this is kind of what we found. So in this plot, I have the designs by either active site focused, which is the top row, or non-active site focused on the bottom row. And our metric here is A to G editing efficiency, where of course higher is better. The points with open Black circles correspond to the starting deaminase sequence. This was the kind of moderately active one we generated from a language model. The dark blue points correspond to designs from proseLM that retained activity, and the red points correspond to designs that didn't have detectable activity at this site. So you can see there's a handful of designs, just comparing the dark blue to the open circles that improved base editing, both from the active site focused designs as well as the non-active site focused designs. And you can also see, if you look at these purple markers, which are a set of base editors from the literature that have been designed through 7 or 8 rounds of directed evolution, we can actually take this moderately active base editor in a single round of optimization, boost its editing to be pretty comparable with some of these highly optimized editors, both through the active site strategy and the non-active site strategy. To give a sort of flavor of what these mutations look like, here I'm showing the 6 mutations to the active site that yielded the largest increase in editing efficiency. This is just a model where I've taken the putative homology model structure and mapped the mutations onto it. So you can't really glean any atomistic understanding of how these mutations are changing the function of the base editor. But I think just by looking at the mutations, for instance, this K69P mutation, this Y, I guess that shouldn't be 69, this Y to R mutation, these aren't kind of modest mutations. These are the proseLM model significantly changing the characteristics of particular residue positions in the active site, which are yielding this large increase in editing efficiency. Similarly, for the design that was directed at non-active site positions, here I'm just showing the positions that were mutated in the best design, and you can see the red spheres are distributed all over this protein dimer deaminase. So again, these aren't modest changes to the protein. These are significant redesigns that are yielding these increased activities. For the final portion, I'm going to switch over to modeling antibody interactions, where here we took a similar approach and trained a model special for antibody design, and then we used it to optimize and diversify therapeutic antibodies. To motivate training a special proseLM model for antibody, here I'm showing another perplexity plot, this time on a data set that I built from SabDab. And specifically, if you look at the light blue line, this is when we provide proseLM models trained on all of the PDB, just the antibody, the perplexity. And then the dark blue line is if we add in the antigen. So it's now a bound complex. And you can see, similar to before, as we increase the scale of our proseLM models, we see lower perplexity in both of these cases. So these models do work for antibodies. They can take in the antibody, assign low perplexity, and then they can further incorporate the bound complex to narrow the scope of the sequence design more. But we also tried taking this ProGen2 model that was trained just on natural antibodies, ProGen2 OAS, and if we then train this instead of training the adapters on the whole PDB, if we just train it on a set of antibodies from SabDab, we can actually achieve much lower perplexity, both for the antibody alone in this light purple dot, and then for the antibody antigen complex in this dark purple dot. And we can achieve this with far fewer parameters than the largest proseLM model trained on the whole PDB, despite having lower perplexity as well. We also looked at sequence recovery for this model. And again, the result is similar. This model that we called proseLM AB achieves the best or close to the best sequence recovery for both heavy and light chains, with or without the antigen, compared to proseLM models that have seen the whole PDB and have more parameters. Breaking this down by different regions of the antibody variable fragment, these collections of bars correspond to, first, the heavy chain framework. So these are the non-binding, more structural regions of the variable fragment heavy chain. Then the 3 CDR loops of the heavy chain. So these are the loops that are more variable and contribute directly to antigen binding. And then likewise for the light chain with the framework there, and then the 3 CDR loops of the light chain. We see the biggest improvements from the antibody-specific model, which is the purple bar for the framework. So these are the conserved regions. We're likely seeing the most improvements here because this model has been exposed to many hundreds of millions of natural antibodies. So it's really picked up the patterns in these framework regions, whereas a model trained across all proteins might see fewer examples of antibodies proportionately. And then for CDR loops, the proseLM AB model tends to be close to the best performing in terms of sequence recovery, though these numbers are a bit noisy because the loops are quite short in some cases. So to test this model, we wanted to look at therapeutic antibodies. Benchmarking models for antibody optimization is hard because there's always a concern of data leakage, and we don't want to set up a design challenge where we can just reproduce something like the CDR loops of nivolumab in this case, and have that look like a success. So instead, we took a therapeutic antibody, and then we tasked our model with finding mutations that would make it better. So we strictly need to deviate from the ground truth in a way that improves affinity. So we split this strategy up into CDR mutations or framework mutations, and tested about I think about 40 for each strategy. And in general, we found that mutations to the CDR loops retain binding about 25% of the time, where about 95% of mutations to the framework residues retain binding. But despite these kind of differences in hit rates, for both strategies, we found a wide range of binding affinities. On the plot on the right, I have the negative log of the KD, so higher here means higher affinity. And you can see that we found a handful of designs from both strategies that had higher affinity than nivolumab for its PD-1 antigen, including two variants, one from each strategy that had about threefold higher affinity in this case. Zooming in on just one of those examples, here I'm showing the CDR variant that had that threefold increase in affinity. This was a two mutation variant where we had one mutation in the CDRH1 loop and another in the CDRL3 loop. These mutations are interesting because the H1 loop mutation actually ablated binding when we tested it in isolation. And the L3 mutation actually only had a small impact towards improving affinity when tested in isolation. So it's really these two mutations in combination that yielded this threefold increase, which kind of highlights the ability of this model to maybe find synergistic mutations that you might not find from a deep mutational scan type approach if you wanted to increase the affinity. Next, we looked at this other therapeutic antibody, secukinumab. I'm just showing the structure here if it bound to IL-17A to highlight this very long CDRH3 loop that you can see kind of extending up the center of the structure. I believe this is an 18 residue CDRH3 loop, so much longer than nivolumab, likely a lot more flexibility in this loop, perhaps easier to break with design. So we wanted to see if we take this maybe more challenging design setting and let proseLM redesign sort of any position in the variable fragment, can we still get binders that bind IL-17A? So we did this by virtue of the conservation of the framework residues. Most of the mutations that we found amongst designs were located in the CDR loops, particularly this long CDRH3 loop. But despite the heavy mutations of the CDR loops, we did find two variants. The one on the left with 31 mutations, shown as the red spheres here, and the one on the right with 18 mutations, again with red spheres. We found two variants that had about 100 nanomolar binding for IL-17A. This was exciting because hopefully you can see that we are significantly changing the CDRH3 loop here, which is making all of these contacts with the antigen. So this really shows that we can perhaps not design sort of therapeutically relevant binding affinities, but we can completely change the CDR loops, which could be useful if you have maybe a starting antibody in a local fitness optima that you need to kind of jump out of that sequence, that nearby sequence space, and then find something that you could then again optimize over multiple rounds of design. To wrap up, just want to kind of recap what we talked about. First was showing how we can do parameter efficient adaptation of language models to enable them to take in structural information to constrain design. In particular, we can provide functional context, things like surrounding proteins or nucleic acids or ligands, to gain really precise control over the generated sequences that come out of a model like proseLM. And then by doing so, we now have a model that we can use to optimize complex functional proteins like genome editors, as well as optimize or diversify therapeutic antibodies for binding. Moving forward from here, we're excited to start adapting these models to additionally condition on non-structural information. This could be things like enzyme classes or scalar values like stability and the like. Also, looking towards kind of relaxing this explicit mapping between the conditioning information and the generated residues, where in proseLM, this is sort of one-to-one. For each residue that we generate, we know the structural context that we want it to adhere to. And then finally, we're continuing to design more complex proteins beyond genome editors and antibodies with proseLM, and exploring kind of where it succeeds. And finally, I want to acknowledge all of my colleagues at Profluent that contributed to this work. If you're interested in reading more, we have a preprint on bioRxiv, and we're planning to release the code for this model publicly in the coming weeks. You can see the GitHub link here. And then finally, if you're interested in this work, we're also hiring across the roles that I have mentioned here, and I'm happy to follow up with anybody about that too. Thank you.
Awesome. Thank you so much, Jeff, for that talk. I think we have one clarifying question just as a follow-up from the talk, and then we can move on. So if people want to think about unmuting and coming up with questions, that would be great. So the clarifying question is can you talk a little bit about how binding affinity was measured?
Yes. So for the antibodies, we measured binding affinity by SPR. We I believe ran SPR with a cellular lysate against the antigens.
Great. Okay. And then one more clarification is, how is the functional context provided as input?
Yeah, so I didn't talk much about that, but I can expand on that. So the functional context is represented as atoms in a similar kind of nearest neighbor graph formulation as we use for the protein. We kind of have multiple graphs interacting here. So if you can see my mouse, this atomic graph is the nearest neighbor graph over just the atoms in the context. I think the K we used here for nearest neighbors was maybe 8 or 16. So it's smaller than protein, a smaller kind of receptive field. And then we have another set of edges that connect the protein residues to these atomic graph nodes. So we have information flowing from each atom in the context to the nearest protein residues, but not vice versa, importantly. Does that answer the question?
I hope so. I'm going to let, if someone wants to chime in and ask anything else, I'll leave a sec for that. Okay, if not, people can feel free to unmute or post in the chat and I can read it out for them. Okay, let me see. Okay, Rakesh, go ahead. I think you should be able to speak now.
Hi, this is just following up on the functional context question. What I was asking is like, so let's say you have a structure, right? So, and maybe let's say it's either interacting with another protein or some ligand. And so how do you like make the model understand that you have to just focus on those set of parts where the ligand interacting residues or protein interacting residues. And those are the places where it has to both locally focus as well as design a global sequence, taking that into account.
Okay, yeah, I think I understand. So we have a protein graph that we start with, which is just the protein backbone. And in that case, we have kind of a one-to-one mapping between each residue in the protein graph and each residue we want to decode from the language model. So when we provide the model to the conditional adapter layers, we just kind of directly provide the residue in the structure with the residue we want to generate. When we expand the protein graph to include other complexes, we essentially use those to add information to the protein chain that we want to design, but we don't actually give those additional nodes to the language model. It never sees those, so they only go so far as to inform. For instance, in this case, this blue sphere would collect information from this gray protein that we aren't designing, and then this blue sphere would go to the language model itself. And then we can do the same thing for non-protein atoms, where those atoms can kind of inform the representation of the nodes that we're going to be designing sequences for, even if they don't go directly to the language model.
So basically you mask out the structure of the binding regions of the protein and then try to predict the sequence, recover the sequence.
Yes, that's right.
Okay, thanks.
Great. Okay. So Pradeep asks, what is your biophysical interpretation of sequence recovery?
Yeah, it's a great question. It's a bit hard to answer. Just show this plot. I think there's kind of a mixture of types of information that are yielding these increases in sequence recovery. So if we just want to take the model trained on structure alone, so the gray boxes here, you can kind of see the direct impact in sequence recovery when you provide something like perhaps an ion is the most straightforward. So you can see just having like the precise backbone information of the ion coordinating residues actually lets you guess what they should be about 50% of the time. And then maybe the specific biophysical properties of the ion itself boost that to close to 70. So there is some information about the ion itself that's kind of yielding increases in sequence recovery. But an interesting trend here is that if you take the largest proseLM model, even without knowing what the ion is, it can already get close to 80% sequence recovery on these residues near the ion. So there's kind of a mixture of evolutionary information from knowing about lots of diverse proteins and the specific kind of coordination around that particular ion that yield increases sequence recovery. It's hard to know if the model is pulling out, or more so the balance of how much biophysical information the model is pulling out from these structural environments versus kind of memorizing this configuration looks like it should interact with this ion. I think the protein MP9 paper actually, or maybe ligand MP9, goes into a lot more analysis of how these things evolve with noise as well, which might shed some light on kind of the contributors to sequence recovery here.
Great. Okay. So Karthik asks, when determining mutations to the antibody, does the model take in both the heavy and the light variable regions at the same time to create log likelihoods for suggesting and ranking mutations? And then maybe clarification on, are they actually log likelihoods?
Yes, so let me show maybe this. So we do use log likelihoods or perplexities in this case to rank sequences. And then to the question of does it take in the heavy and light chain variable regions at the same time? It does not in practice. So what we do is we have the structure of nivolumab, and then we can generate the heavy chain either by conditioning on the full FV structure, so heavy and light chain without any sequence provided. That lets us kind of start the generation process. Then once we've designed a heavy chain, we can then design the light chain condition on the full structure again, but this time giving the heavy chain sequence. So then we can generate the entire sequence given just the structure. And then to score these, of course, the order of heavy and light chain is arbitrary, so we score both ways, either like heavy chain given light chain, or vice versa, and just average those. And then we select the sequences based on perplexities from a panel of proseLM models. So in the bottom row of this plot, I'm just showing the perplexities of the sequences we selected for CDR variants, or I guess these ones are framework variants, versus the entire set of sequences we designed in gray.
Cool. Okay. So another question is, what would be the computational resources for fine-tuning proseLM on specific classes of proteins? I don't know if there's like a specific metric that they're looking for, so feel free to give some more detail.
Yeah, I can maybe answer generally. So all of the models that we trained here were trained on a single A100, and it typically took for the smallest models on the order of hours, and I think for the largest proseLM model trained on the whole PDB, that one took like 3 or 4 days, if I remember correctly. So in the grand scheme of things, pretty cheap relative to training the language model itself.
Awesome. Okay, so follow-up from Karthik, I think, is there a difference in how you use the model for suggesting single and multiple mutations to an antibody?
Yes, there is. So the difference between the two is for the CDR loops, I actually exhaustively considered every single or double mutant that could be made in those positions, excluding things like mutating to cysteine or proline. So that was a set of about 500,000 variants, I believe, that we just needed to score all of. So we could enumerate every single combination and then score them and pick the best ones. And then for framework design, where we might be changing, let's see. Yeah, we might be making, say like 10 or 15 or 18 or 19 mutations. There we can't exhaustively consider every sequence that could be enumerated. So here we do this fixed position design where the CDR loops are provided to the encoder, and then the language model just generates everything. And these are kind of the observed distributions of mutations we got.
Okay, someone's asking about, can you use the model for nucleic acid mutations?
As it stands now, it only generates protein sequences. There are nucleic acid language models. You could do something similar with those, where you take the small set of nucleic acid structures you have in the PDB, and then build conditioning into something like a DNA or RNA language model. I think that would work. If the question is more about like changing specificity for nucleic acids, I think that is more in scope for a model like proseLM. One tricky part might be if you are mutating nucleic acids that are kind of evolutionarily conserved. The language model component of proseLM is then kind of competing with the structural component. So in those cases, it might be useful to just use like the encoder itself to get a more pure structure-based approach that might be more receptive to those mutations.
Great. Okay. So another question is, will fine-tuning of ProGen 2 model first on specific protein classes and then using those embeddings for proseLM improve sequence recovery?
Yes, I would expect so. It also kind of reduces the amount of data you need for training the proseLM adapters. That's kind of the case with the antibodies, where we already had this model trained on the observed antibody space. So I think about 500 million natural antibodies. And then to build it into proseLM we just needed to take that model, take the encoder that was trained on the PDB and train an adapter between them.
Well, okay, so maybe this is a good final question, but someone asked, do you have an idea of when the coder model will be publicly available?
I'm hoping within the next few weeks. Still working on cleaning up the code, but our plan is to put it up on this GitHub link. If you do want to see something like the code earlier, I tried to document in pretty good detail the methods and the supplement of the paper too, with pseudocode.
Okay, awesome. Seems like people are excited to play with the code when it comes out. But okay, great. Thank you so much, Jeff, for today's talk. The recording will be online probably by the end of next week, and you can always go ahead and catch up on there. And with that, we'll see everyone back in 2 weeks, and we're going to hear about Cheap from Amy Liu.